So, good morning, everyone. So, today we'll be starting with our next lecture that is on denoising. In the previous lecture, we performed demultiplexing of the sequences and we got certain files which we analyzed and we checked. We also got certain interactive visualization files where we got a box plot bar chart where we analyzed each and every read, what is the quality score for that particular read, and then we can decide from that, as I mentioned, that okay, till where do you have to take the sequence and from where you have to truncate the sequence. That is, you don't need to carry that sequence forward because the quality score has been reduced. So till where we have to take it, that and all we understood from the demux.qcv file that we created last in the last session, and we did the analysis. So after demultiplexing, the next step comes is denoising. So I'll share my screen and let me know if it is visible. Is it visible?
Yes, ma'am.
Okay. So today's topic will be about the sequence quality control and denoising. So as I mentioned before, that when we proceed further with the sequences, what happens is that there might be certain errors or an unwanted sequence or short reads that are present, and some sequences could have a low quality score as well. The way we mentioned in the last session when we checked that bar plot, I told you that the bases after 100 show a very low quality score. That means that from there the sequence, the basis interpretation or the prediction cannot be taken very seriously because the quality score is very low. The system itself is not sure that is that piece present there or not. So just in case, to avoid any misunderstanding or any misinterpretation, we generally remove those part of the sequences. So that removal is one of the part and one of the step of sequence quality control that happens in a denoising step. So now today we'll be studying about the denoising step, and we'll be running the denoising step command so that we'll get an output of how the sequences look. So basically for demultiplexing, we used to get that interactive bar plots and all of that. But for denoising, it's just going to be one table that will be telling that which and all reads we retained and how many of them were removed and total percentage of the reads that were like retained after doing all the analysis. So here, after doing the sequence quality control, then the later parts come is feature table construction, where you will be just putting the feature and how much reads are supporting that thing is going to be captured, and that will be represented as a table. So today we are going to do the denoising by using data 2. There are two tools that can be used for denoising according to our moving picture tutorial, that is data 2 and dblur 2 and sorry dblur. So, but in our presentation and in like if we go further, I'll be teaching only data 2, and both of them, data 2 and dblur, are similar, but data 2 is more flexible and gives like more accurate results compared to dblur. So that's why we are focusing here. And dblur will be a homework. So after we are done with this session and the next session, you already have that kind picture tutorial link, right? I have put it in the
Yes,
lecture
ma'am.
third slide. So open that and you can check for the commands there. And after today's session, in the next session, I would like to see the outputs that you got from dblur after performing it on your own. So today we will perform data 2 in the class and dblur will be a homework. So I will be introducing data 2 that is used in kind 2 and how it is used to analyze and how does the command works. So first of all, why is quality control needed? The main reasons behind that is that there might be certain errors in the bases when the bases are sequenced. There can be certain regions that are low quality, that is the base quality scores are low for that, and those bases cannot be distinguished very properly. And there can be reasons that there are certain chimeras, that is some artificial sequences got generated during the PCR amplification that led to the generation of certain chimeric sequences, which will hamper our further analysis step. So all these errors, when they combine together, they can affect our phylogenetic analysis later. Obviously, if there are chimeric reads or there are certain error reads that contains error, so when we are sequencing and analyzing that, that can lead to its interpretation in a wrong way. Say if it was supposed to belong to a kingdom, say it was supposed to belong to a genus Bacillus, but then it came to Streptococcus just because of a certain region mismatch. So that is a complete uh, wrong analysis that we did, because the bacteria was supposed to belong to Bacillus, but it came to a Streptococcus. So to avoid all of that, and then to avoid any diversity analysis problems, and to place them in a correct taxonomical class, these and all errors should be addressed beforehand. And also in the later steps, the diversity analysis can also be a problem. Say it was a bacteria, you're doing a microbiome study of two things, one from one sample and one from one sample. But because of these errors, those samples sequences are not well defined and well differentiated. So there will be confusion that from what sample did this sequence in coming, and all of those will collectively lead to an improper diversity analysis as well. So all in connection, these things are very important to be removed, because errors, these low quality regions, these chimerics, these PCR duplicates, they will cause a very big problem later on when we start with the analysis part later. So just to be like on the correct side, we will overcome all these errors, and we will remove it, so that we get a proper biological result analysis in the later parts. So, what is denoising specifically? So, denoising specifically is that you are removing any unwanted sequence or any unwanted bases that are present. Say you are cleaning a room. So, here in biological reference in microbiome data, when you are doing cleaning of the sequencing data, then they have given it a specific name that is called as denoising. So, what will denoising do? It will remove all the sequencing errors that are present, and it might correct the incorrect basis. So what will happen when it is going to remove the errors, then the basis will obviously be corrected on its own. It's like a dependent process. And after the correction that happens, and obviously the reads of the sequences that will be produced will be of high quality. So what happens is, once the sequencing bases are removed that have errors, then those resulting sequences are high quality resolution sequences, because they will have no more errors further, and those can be directly processed for downstream analysis. So after denoising, what happens is, instead of getting a clustered sequence, say clustering like similar sequence in one, and removing the non-similar ones, what will happen is it will give an exact biological sequence. So now these high resolution exact biological sequences are called as amplicon sequence variants, that is ASV. I do remember mentioning in the initial sessions about OTUs and ASVs. OTUs are the operational taxonomical units, and ASV is the amplicon sequence variant. So in OTU, what happens is that uh, you cluster all the sequences that are similar to each other in a one group, and the ones that are not similar can be is sent to generally either discarded or sent to another group that has a similar sequence. But if those number of those particular sequences that are not matching the larger group, if they are very less in number, they are generally discarded. So what happened here is, in this denoising, we are not making clusters, that is, we are not making OTUs, we are making ASVs. That is, instead of clustering sequences together, you will generate each and every single sequence separately that is corrected of any error. And those sequences are known as amplicon sequence variants. So after denoising, what will happen is that the all the noisy reads or noisy data is converted into a relevant or an accurate biological sequences that can be processed further. So now what is data? So data is basically an error correction algorithm that is used in this particular kind to pipeline. So it generally works on a Illumina amplicon data. So here, as we are studying the amplicon sequencing, amplicon is basically the short stretch of sequencing of entire genome. Here we are not approaching the entire genome, and for that particular thing, this amplicon sequencing, the data too is considered to be more beneficial and more used, because it is flexible and gives high resolution analysis. But when it comes to the the entire shotgun sequencing that is happening for the entire genome, then there are multiple other tools that you can use. There you can use deblur as well, but for amplicon, data tool is generally preferred more because of its flexibility. So now what happens is, so It is basically an advanced algorithm. Okay, so now it works specifically on Illumina sequencing, I already mentioned. And now these are the steps how this internally works. So here you just write a command data to, and then it will process and give you the final result. But that generally doesn't happen like that. There are certain internal steps through which the algorithm works, and each and every step after it is accomplished, then it moves to the next step. So the initial step here is filtering. So it will filter the low quality reads. First, it will check. So from where it is checking? You generated a demux.qzv, right? In demux.qzv, you knew, you can see that which bases have a low quality base, sorry, low quality score. So from based on that information that is present there, it will filter out all the low quality reads, and then it will learn the error rates like from the data. So it will go through the data. First of all, it is going to do modeling itself. That is, it is going to check what and all errors are there. Then it's going to model. Maybe it will have certain previously mentioned data as well, because it acts as a model, and that data acts as a data for training this particular model. So after, it will learn, after the process of learning the errors, that the algorithm will learn itself, and then it can implement that for a particular data. So now it will go and move on to denoising, where it will go and correct all the errors that are present based on the previous error detection rate. And then it is going to go and check if there are any chimeras, or if there are any sequences that got generated during PCR duplication and all of that. And then it will remove all of that, and it will also remove the denoising data. So on whatever step we are, it is going to document that step. Say if you come from filtering, so he has filtered say 10 reads, so that will be documented in a table that we will be analyzing. And then if say denoising, in denoising step it has removed 20 reads, that will be documented there. And it has removed 70 chimeras, so all these columns will be there. That is how many reads are filtered out, how many reads remaining, all of that. And eventually it will show how, out of the total number of reads that we had for one sample, how many are remaining after doing all these pre-processing steps. So now, as I mentioned about OTUs and ASVs. So when we use this, is we are using KIME 2, right? In KIME 1, what they used to do is they used to generate these OTUs, that is operational taxonomical units. So in OTUs, as I mentioned, what happens is that you check for sequences. So there are two sequences. And then when there are two sequences, and if they are more than 95 or 97% similar, they will be clubbed together. See, it is 97%, right? And in 97%, what happens is that say there might be certain errors in a particular sequence that can happen, and those are generally disregarded here. So that is why there might be certain chances of error still. So that's why like this OTU has become like overrated now, and that's why we have moved to amplicon sequence variants. So that is how there was a shift that happened from KIME 1 to KIME 2, where we shifted from operational taxonomical units to amplicon sequence variants. So on the other hand, what happens in amplicon sequence variants is that instead of clubbing them together, here they generate exact sequence. That is, they will check one particular sequence, they will see if there is any error or not. If not, they will just move it to the next part. But if there is any error, it will correct. It will not be based on any relevance with somebody else or similarity. It will work on an individual basis. That is why in amplicon sequence variants, we get generally 100% exact sequence, which has higher resolution, which are more accurate, and it will detect even if there's very small differences that are present between organisms. So that is why data 2 is much better than the OTU based methods, because you can differentiate on a very cellular, on a very base level basis. That is, if there is any change in one base or two bases, that can also be detected here, because here we are generating 100, here we are generating 100% exact sequences. So now when you perform the data, this is all what how data to works and what is the mechanism behind it. And when you do basically like do the data to analysis, so you are supposed to get some files. So these are the four files that we'll be generating when we perform data to. So it will be named under the same things. One will be stats data2.qza. So this will basically contain all the statistics. That is how many reads were filtered, how many reads were denoised, then what were the non-chimeric reads, how many were the chimeric reads. So all of them will be present, and then we'll have a numerical value for that. That is initially how much reads we had, and then finally how much did we get. As this is a .qzf file, it is difficult to visualize. So that's why we'll convert this stats data to file to a .qzb file where we can analyze it in the browser. Okay. And then after that is that we'll generate one other file that is a table data2.qza. So what will it do? It will be a feature table generation. So it will count each sample. So it will count each sequence in a sample, and basically it will tell, okay, for this particular sample, these many sequences are there, or vice versa. So this will be a dat file or a data file, and this will be a representative sequences data to file. So what happens is when we are doing that ASV, so I told, right, each and every single sequence is generated. So here it is telling me the feature count, but here it is directly going to give me the sequences itself, that okay, these are the DNA sequences that we are working with. So these two files are not much of importance in the denoising step because these are the byproducts. These will be used and analyzed further when we construct a feature table in the next session. So right now today we will be generating all the 4 files, and we will be visualizing the stats data.qzb file and check what and how the data comes after denoising. So now I have been constantly talking about the filtering part. That is, the first step is filtering. We filter out reads and all of that. So on what parameters are we filtering out those reads? So basically there are two parameters. One is p trim left, that is it will remove the bases from the start, and p trunk length, that is it will cut a sequence at a position. So when we say p trim left, so what happens is, say from the start, it starts from base 1 and it goes till base 150. So initial 10 bases you don't want because that are not showing good base quality scores, or those are adapters, or those are not included in our sequence proper. So we will give that parameter here for p trim left, and then we will trim those initial starting bases. And then p trunk length is that at any position that you want, you can cut the sequence. So when we had a 150 base pair ka sequence, we looked in dmux.qzv in the initial sessions. So what happened was that after 100 bases, we saw that the base quality score kept on reducing. So we are not interested in the sequence where the base quality score is reducing, because it might give me unnecessarily opposite results that I'm not interested in. So what I'll do is I will put the truncating length as 100. That means at 100 position, my sequence will get cut, and after 100, I will not be getting, only from 1 to 100 base pairs, my sequence is filtered out. So that is how you set parameters to cut the sequence from the start and cut the sequence from the end. So it's not just ending, it is just that anywhere you put, even if your sequence is 110 base pairs, you put at 10. From 10, the sequence will get truncate. You will get 1:10 base pairs, and from the later part will be in the next sequence. That is, you cut the entire sequence into 2 parts. So that is how the truncating command works, and this is how the initial basis sequence removal command works. So now, how are we choosing these parameters? This, I hope that this should be clear beforehand itself. So what happened is, this is how our graph looked, right? For our bases that we got in dmux.qcv, right? Do you remember?
Yes, ma'am.
Chaita and Mandita. Yeah. So it was supposed to be like this, right? Initial basis had very good quality score, but as we came towards the end, here I have just mentioned an arbitrary length that I took just on my own. That is, as we go on to the next step, what happens is that the sequencing quality score is getting reduced. So initial bases are high quality. I don't want to trim anything from the start. So I will keep the trim left as 0. Okay. But as I go on further, here we can see that after approximately 120 or 100 base pairs, my sequencing score is getting base quality score is getting very low. I don't want those bases anymore. So I will put the truncating length as 120. So after 120, nothing will be considered. From base 0 to base 120, my sequence will be considered and taken up for the further steps. But after 120, it will be dropped because the bases are not good. And that's why I truncated my length till 120. I don't want the bases after 120. So these and all information you will get first when you analyze dmux.qcv. Without that, you will not know, right, till what sequence you need to truncate or till what sequence you need to perform the denoising. So that is why all these steps are connected, and that is why you should understand each and every step. Then only you can move on to the next step. So this was the input of the dmux.qcv that we generated in the last session. So now we'll start performing the data to analysis. So you guys should be ready with your terminal, I will send this command. Any one of you again share your screen. I would like to see how it is working for you guys. I have pasted the command in the chat box. Uh, any one of you, Chaitra, share your screen this time, like always Manvita does. So this time maybe you can share your screen.
Okay, ma'am.
Okay. So open share your screen and just copy this command. Manvita, in the meanwhile, you can run this command and tell me if you get the output or not. So what are the prerequisites for running
Okay,
this
ma'am.
command? Yeah, the kind tools generally take a little time to run because these are a little big commands. So you can see here, right? What is the length that I'm trimming it at? Initial basis.
Yes, ma'am.
So I've like when you look at the command, can you guys figure out that from what to what I'm trimming?
Like, ma'am,
Hmm?
left trim is started from 0,
Okay.
and final length is 120.
So when I'll
Like
get my
cutting
product,
the.
yeah, correct. So when I'll get my product, that is after trimming everything. So what do you think? What will be the length of that particular sequence?
Ma'am, 120.
Yeah, good. So let's wait till the time it runs. When it's done running, just inform me. Then we'll check for the files that are generated.
Okay.
Okay. So like just look at my screen now. Is my screen visible?
No, ma'am, I will.
Yes,
Your
Manvita.
meeting screen is visible. No, 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 your meeting screen. Now PPT is visible.
Okay, okay. Manvita, you were saying something?
Now it's visible, ma'am.
Okay. So your commands are running in the meantime, right?
Yes, ma'am, it's still running.
Okay. Just let me know when it's done so that we can analyze the files that have been generated. So now, when we ran this command, no, now it is taking time to process, right? So what is happening inside? So first step it will go on is that is it will filter the low quality reads. That is, we have put the parameters, right? That we want only from 0 to 120. So it will filter out all the reads and it will only select those that like only keep those that are from 0 to 120. If there is any reads that is smaller than that or something, it will obviously remove it because that doesn't fit into a filtering quality reads criteria. So after that, it is going to model the error rates based on the sequences and the data present. And after that, it will implement that on our particular sequence and it will correct the basis or the sequence. So if there are certain things that will not be corrected, then it will just remove it out as well, and then it will check for any chimeras. If there is any chimeras or any artifact sequences that is generated using a PCR duplicate, then it will remove that as well. And then finally, it will generate an ASV that will be present in a file that we know that will generate different files, right? One will be the statistics file, one will be the feature table file, and one will be the sequences file. So that will have our output ASV sequences, and that will be like a proper corrected sequence for each and every sample. So now what happens is we generated a file, right? That is stats data2.qza. As this file is not like in a visualization format, that we cannot see. So what we will do is we will convert it into a QZV file so that we can visualize it. So I'll copy the command in the chat box again. Did the command run, guys?
No, ma'am.
No, ma'am, it's taking too much of it.
Yeah. Okay. Because it has to undergo so much parameterization, that's why it's taking a little time. And it depends on the system as well. So let it run. After this command runs, then you will be seeing those files that I mentioned in the presentation. Those files will be generated. These three files will be generated. Okay, when you will see in your folder stats data.qza, table data2.qza, and rep seeks data2.qza. So all these files will be generated, and then what command I just copied pasted in the chat box will be used to convert this data stats data2.qza into stats data2.qzb, so that we can visualize that particular file. So now why are we converting it? Because obviously the reads are retained, the reads are filtered, and chimeric removal. These are all things will be shown in the QZV file that we can visualize very easily as compared to .qza. Because QZA is basically not a human understandable format, it's a compressed artifact file. That's why we need a visualization file to see all of these things. So now as we are moving further, so they have replaced, because they in the moving tutorial thing, they have performed the sequencing, this denoising, once by data2 and once by dblur. But then they are further taking it as a normal. They have normalized the name, and then they're taking those outputs. So I have also renamed both the files that we will be using further. So these are the two files, that is the sequencing files and the feature table files. So these are the normal files. Like I have removed data2 because these are the nomenclature that have been used further. So just to prevent any conflict and all of that, any error in the commands, so I have just renamed it. So after running all the commands, you also guys rename the file that are generated to repseeks.qza and table.qza, that we can move further. And there shouldn't be any problem with the commands, and because I will be putting these names in the further commands, not the data2 things. So now what outputs we got is, first we got that high quality sequences, right? The ASPs, that each and every sequence is properly corrected, and you 100% correction happens, and then it is presented in an individual manner where the quality is really high and the resolution is also high. Then we will be generating a feature table where the number of reads or sequences per sample will be mentioned, and these files will be further used for the feature table constructions. Now next is, now these outputs that are generated can directly be used as a taxonomical analysis as well, but we'll be converting it to a feature table, and then we will be processing with the taxonomical and the diversity analysis part. So this I mentioned before. So dblur, you can use dblur as well instead of data2, and that is your homework for the day. So in the next session you should show me the files that you got generated using dblur, and you need to run the commands similarly the way we ran for data2. It will be just a dblur plugin instead of data2, and it was present in that moving picture tutorial thing. Just go through it once and understand what dblur is. In the next session you guys should tell me what dblur is and how it works, and what are the files that you generated. So these are both the similar things, but one is more flexible and one is more faster. So both have their own pros and cons. But the outputs are very similar because it also works in a similar manner. So this was all about data2 today for today's session. So did
Ma'am,
the command, yes?
ma'am, can we run the second command?
Oh, did the command run? Can you just share your screen and show me what files you got generated?
Sure, ma'am.
Manvita, is it done for you as well?
No, ma'am. It's still running.
Okay. Yeah. List all the files, Chaitra.
Ma'am, table data to dot
Yeah,
q.
just do
Oh,
ls. Yeah. When I say list all the files, you have to just type
Okay,
in the
ma'am.
command.
Okay.
So see, you can see here the files that are generated. That is stats data 2.qza, table data 2.qza, and repseeks data 2.qca. These are the three files that I mentioned, right? That will be generated.
Yes, ma'am.
Okay. So the stats will have all the feature table numbers, sorry, stats will have all the numbers that we'll be looking now after we convert it into .qzv file. Table will have the feature table counts, and rep6 will have all the ASV sequences. Now you run the next command, that is to
Okay.
convert the QZA file into QZV file. Okay. In the meantime, it's running. You can stop sharing your screen. I will show you guys the output. That is how does the stats data 2.qzv file looks. Okay. So this is how it should look. When you perform everything and when you get the .qzv file, you will upload in that viewkime.org website, and then you will see this sort of a file get generated. So here you can see that you have the sample IDs. These are the total 34 samples that we have been looking at since day one. And then what are the input? Input is the total number of reads that you input are present for these particular samples. So when you did denoising, when you started denoising, so there were total 1130 reads for the sample 1. Okay. So now you put all the criterias, you denoise, you did any chimeric analysis and all of that. So now what happened is after putting the filter where we only took the sequences from 0 to 120, so only from 11,340, only 8,571 reads were retained. So that is, when you subtract this from this, those were the total number of reads that were discarded because they didn't fall into our filtering criteria. So after filtering from 11,000 reads, we only got 8,500 reads. Okay. So the number of the percentage of the reads that pass the input filter is 75.58. So whenever this percentage is greater than 50, it is a good result. So these results are a little 47, 40, not that promising because only very few reads were retained from these samples and rest all were discarded. So this is basically the total percentage of the reads that was able to pass the input filter, and the 0 to 120 base pair sequence was retained. So when you performed denoising on this 8,571 sequences, you saw that only 8,499 sequences were retained. So all the other few sequences that were present were removed in denoising because they had some errors and all of that. Now we input the 8499 sequences to check how many chimeric reads are present. And then we saw that total 7780 reads were retained that were flagged as non-chimeric. So from 8499 to 7780, what is the difference? One second. So when we subtract this, we know that there are 719 reads that have been removed because those were flagged as chimeric reads. And out of total, 7780 are the reads that have been retained. And these reads are the ones that have been already filtered. They have been denoised and they are the non-chimeric reads. So if you see the total percentage that is out of this, so from the total reads, 11,340, we did all the filters and all the parameters check on our reads, and we got only 7780 reads in the end. And this is 68.6 reads of the total reads. So now from here, we got to know that, okay, total number of reads that acted as an input, how many reads were filtered during the filtered process, then how many reads were retained after denoising, and how many reads were retained after doing the chimeric analysis. So this is how your file looks for the stats data 2.qcv. Only one file here is generated that we needed to visualize after denoising. So we got to know that how much percentage of the total reads is retained. So 68.6% of the reads have been retained from the total number of reads. So for each sample, it has been mentioned, and this is what we analyze using data to analysis. Other two files are the QZA file, that is artifact files. We will be directly using them to generate feature table, and then we will be visualizing what results we get during the feature table visualizations. So I hope it is clear how does the data to command works and how does the outputs look like.
Yes, ma'am.
Okay. Did your files run? Did your command run?
Yes, yes,
Yes,
ma'am.
ma'am. We got the output files.
You got the .qzb file as well?
Yes, ma'am.
Okay. So like upload it in that view.org, and you should also get the similar file. Just upload it simultaneously when we are on a call, and so that if it doesn't come like that, then we can work on why it didn't come, but it should come like that exactly. So just upload it and just let me know. And that next command is very important. I've copied in the I've pasted in the chat box. That is the renaming command, because if that rename hasn't happened, then the next command that I will give, it will not work properly and it will show error. So before you guys end up with the session, so just rename your files once, okay? And just show me the outputs once after uploading it into viewkime.org.
Okay, ma'am.
I'll just stop sharing my screen. You guys can just share it one by one and show me. Yeah, that's great. You can also see the similar outputs, right? So this is what we were expecting. And I hope that's clear how to analyze this output as well.
Yes, ma'am.
And Manvita, you also just share your screen and show me the output once.
Yes, ma'am.
Yes, that's the correct output. So these were the outputs that we were expecting after doing denoising. I hope that why we are doing denoising and how is the output, like all of that part is clear. Do you guys have any doubt in the commands that we run or the outputs that we got?
No, ma'am.
Okay. So the homework is that today's session is getting over early as well, because it was a short session, because denoising didn't like is not a very long process and the outputs are also small. So the homework is that you guys should do the same running of these commands using dblur. And in the next session, tell me how does dblur works. Like you can distribute it among cells. So I want to I want to know answers to a few questions. That is, what is dblur? How does dblur do the analysis? What are the outputs and the explanation of the outputs? So these are the four questions that I want you guys to address in the next session. So before starting the feature table generation, we'll understand all these things. You guys will be telling me, and then we'll move on to the feature table. If you guys have any doubt or you need any material regarding dblur, but it is like as I've already shared also before in the lecture 3. But if you want the link again, just text on the group. I'll be sharing it again. So that's it for today's session. If you guys have any doubts, you can just unmute yourself and ask.
Ma'am, like you have sent that last command in chat box, right? What does that command do, ma'am?
Which command, Chaitra?
Ma'am,
The last command.
MV6, yes, ma'am.
Okay.
Data 2.
Yeah.
User.
So what we generated before was, you can see, right, the files that we generated were named as repseeks data2.qza, right?
Yes,
The
ma'am.
file. So we are just renaming that file into a repseeks.qza. We are removing that data2 part because it's a very big name of the file. And in the further commands, I have used just repseeks.qza. So it is just renaming a particular file, nothing else, not a very big command, just renaming command. So that the commands become a little more simpler in the next time because the names of the files are very big otherwise. That's it.
Okay, ma'am.
Okay. Any other doubts, guys?
No, ma'am.
Okay. So you understood, right, your assignment on Deebler?